in this segment, we're going to go over the viral replication cycle that is described in chapter 13. So, what I really hope that you'll be able to do at the end of this is understand how the viruses replicate and also compare that to something like the replication of HIV, a very specific cycle that has some variations over the general cycle. So, just remember that the virus is very dependent on the host cell to do all the replication. And typically, the replication is going to end up in death of the cell, but it might be a little bit longer term depending on the type of virus. So, in this example, we're looking at a bacteriophage, much like the one that we're working with in the lab. And the steps to viral replication are shown here in bold, and we just basically talk about attachment, how it is going to attach to the cell, entry, synthesis of all of the viral particles, and then, of course, assembly and release. Okay, so if we look at this animation that I drew a long time ago, you can see how this kind of works. So if we're looking at the first step, we're looking at the virus, the bacteriophage, and it has to attach to the host cell, and it does that with the tail fiber, which is the viral ligand, to the host cell receptor. And then in this case, how you're going to get the genome in, the entry part, the virus actually injects first enzymes to break down the cell wall, and then its genetic material, which will pass through the intermembrane channels of a bacteria like E. coli to get into the inside. After the entry, you're going to have to make new viral particles. So you're going to use that genome of the virus to synthesize new components. So you need both proteins of the virus as well as copies of the genetic material. And once you have enough of that, you're going to assemble new viruses. And this is really a spontaneous process. You can think of it like a crystallization that happens if you have a bunch of sugar in water and then you drop something in it like a stick, and then you start to evaporate the water. You'll get a little crystal forming on the stick, and then it all crystallizes out over time. It's a spontaneous reaction. The same thing is happening with the virus. It's spontaneous. They'll assemble into new viruses, which is kind of interesting to think about, because something complex like this, you might think of like, well, you're building all the little pieces of a gingerbread house, and then when you have enough of it inside the cell, they just kind of spontaneously come together and form your little gingerbread house, which is the virion. And then they have to get out of the cell. And depending on the cell type, we'll talk about this in this section, it either is going to lyse the cell open and, all the viruses spill out, or you're going to have a virus that kind of pushes its way out of the cell. It's called budding. It buds its way, pushing its way outside of the cell, and it usually is taking membrane with it, and that's how you're going to get an enveloped virus. So let's take a little bit of a look at this. So this cycle might look a little familiar to you because we were talking about transduction in chapter 7, and this is just a regular lytic cycle of something like a T-even bacteriophage, where the virus enters into the, attaches, and then it's going to inject its DNA. That's the entry step. You're going to drain that infection. You're going to be breaking up the bacterial chromosome as you're making new copies of the virus. Then when you have enough of all those components inside the cell, you assemble new viral particles, which then break out of the cell in a lytic type of cycle. So you're looking at the bottom of all of the components that you would need to assemble a virus like this. And you can go into Mastering Micro and look at some of the animations from chapter 13 about that. So if we look at this graph first, you'll see how this kind of lytic infection is depicted. So if you're looking over here on the y-axis, you're going to look at the intact virions, right? Viral particles that are capable of infecting. And if you start with just a single one, when that enters into the cell, right, you're going to have no infectious virions, because now you have just genome of the virus inside the cell. And if you were to break everything open and look for virus at this point, you will not have an infectious virus, right? You'll have viral genome. So then you're going to have a period of time elapse while the virus is replicating the components it needs and then synthesizing or assembling the new ones. And as soon as you have all of those assembled, then it can break out of the cell and you would have a mass release at one time of all the viruses, which would then start a new cycle of infection in nearby bacteria. So this is called that burst time. It's that time from infection to when all of a sudden you can detect viruses in that media again, and that's to replicate viruses. All right, so at the bottom of this slide, you can see a little animation that's just depicting sort of that spontaneous assembly of viral particles that would be happening inside of a cell. Remember, though, that for some of the bacteriophage, we have the ability to enter into a lysogenic cycle, which is a part of the cycle where the phage will integrate itself into a chromosome. And the viruses that are capable of doing that, we're talking about bacteriophages now, bacterial viruses, they're called temperate phages. And when they go inside the chromosome, you refer to them as prophages. Those are the ones that are sitting inside the chromosome. They've integrated into the chromosome. All right, so an interesting idea, something that actually happens, is sometimes those viruses bring extra genes with them from their previous host, right? A kind of transduction event where they're bringing genes from the previous host into the new host, and that is called a lysogenic conversion, because the phages carry genes that alter that phenotype of a bacteria, and sometimes these things are toxins to the host cell. So for example, if you've heard of the disease scarlet fever or diphtheria, this type of thing happens when the virus that is infecting a bacteria allows that bacteria now to have a new phenotype, and in this case a dangerous one, because it causes a new disease in the human. And it's only because that bacteria is infected with a virus that brought an extra gene along, and in this case toxins. Okay, so keep that idea in mind, lysogenic conversion. In the latter part of the semester, we're going to be talking about some of those diseases I just mentioned and the fact that they're related to lysogenic conversion. This is that figure that shows the relationship between the lytic cycle and the lysogenic cycle. So sometimes when a phage injects its DNA, the DNA incorporates itself into the chromosome rather than entering directly into a lytic cycle. And that integrated phage will just replicate with the bacteria. So every time the bacteria divides, it's replicating the virus DNA as well. And then there'll be some sort of event that's called induction. Sometimes it might be a stress or something that encourages that phage to emerge from the chromosome and then enter into that lytic cycle and complete that infection for that particular cell. Okay, so that's just showing you the relationship between the lytic cycle and the lysogenic cycle, and this is the normal relationship. In chapter 7, we were looking at events that were a mistake, right? A mistake that happened either in this stage, right, which could lead to generalized transduction if you had an accidental piece of bacterial DNA that got incorporated, or at this stage where you emerge from the virus emerges from the chromosome but brings bacterial genes with it, and that was specialized transduction. So these were the mistakes that led to transduction, whereas this particular slide is really just showing us the normal, no mistakes made type of event. If we're going to consider animal viruses, they have the basic replication pathway as the phages, all that attachment and then entry and then synthesis and maturation and release. All of that is really generally the same. But for some of the animal viruses, you're going to have an envelope, which is that lipid bilayer that is around some of the viruses, and it's just membrane that they have stolen from their host cell. We have to keep in mind that eukaryotic cells are different than prokaryotic cells. For example, animal cells have no cell wall. Animal viruses don't have that tail or tail fiber type of arrangement that we were seeing in bacteriophages, but they do have those glycoprotein spikes. So that coronavirus that we spoke of in the last video has glycoprotein spikes that in the envelope that will allow that attachment to the host cell. And that attachment is mediated by the terms what we were talking about. The cell has a receptor, and the protein that's on the virus that is interacting with the receptor is referred to as the ligand. When an animal virus enters, it's using one of three mechanisms that are depicted in this figure from your book. So the first they're calling direct penetration, but basically after the virus attaches, it's using a pore or a channel that's inside, it's on the membrane of the cell, and it's kind of shooting its genome right through that pore. So that direct penetration is using a channel to get in. The other mechanism is endocytosis, and pretty much any virus could use this on an animal cell. So endocytosis, if you might remember from another class, is basically when the cell kind of pinches in a piece of the membrane. So once the virus is attached, that cell is somehow stimulated to pinch that piece of membrane inside. And so now you have this little vesicle that has been incorporated into the cell, and then the outer membranes are going to be dissolved away, and the capsid will be dissolved away by the host cell, and now the genome is inside. The third mechanism is membrane fusion, and that's very specific to enveloped viruses. So some of the enveloped viruses do that. And this is when the two membranes, the envelope membrane of the virus and the cell membrane, are brought into very close proximity so that they actually fuse together, kind of like when you push two soap bubbles gently together and they fuse into a larger one. That mechanism of fusing those two membranes together dumps that capsid inside of the cell. Okay, so those are the three mechanisms of getting an animal virus inside the cell once it has attached. Now, the replication is going to be different depending on the type of nucleic acid inside the virus. So if it's a DNA virus, it's going to go inside the nucleus typically and just use the machinery of the host cell just as it is. If it's an RNA virus, though, it's often replicated in the cytoplasm because most of the machinery is there. However, RNA viruses will definitely have to use their own polymerase. So they're going to have their own special polymerase to deal with the RNA genome, because our own cells don't typically deal with replicating RNA, right? We make RNA from DNA, but we don't replicate RNA. So we have to kind of think about how message is going to be synthesized, what's our template for the genome, etc. And don't memorize this, but this is a comparison chart from your chapter that's kind of comparing all of the different types of genomes and what the cell has to do, or what the virus has to do in order to allow the cell to make message, because obviously the virus is going to have to make messenger RNA to make its proteins, and then also what it uses as a template for a genome. And if it's DNA, then it's pretty typical. of what our own cell machinery is going to do.